So I guess it's 7:30. So we'll start. The others will join as per Indian Standard Time, right? Okay.
Good one.
Okay, thank you. So we'll start the session. First and foremost, I wish everyone a very happy new year, because this is the first session that we are conducting in 2026. And I'm very happy that so many of you have registered for this event and have expressed happiness that we are organizing such an event. Many of you know the purpose of this. Most of them are repetitive participants. We, as a group, Spectratix believes in sharing of knowledge, and this sharing of knowledge should not be limited with respect to any cost or something. So we invite a lot of industry professionals, and it's sort of a discussion, so that we keep ourselves updated with respect to what's happening in the industry. And the topics are also varied. Of course, we don't leave the pharma domain as such, but we try to remain in the broader sense of pharma. Means we try to cover everything related to pharma, nutraceuticals, everything. And I'm very happy with today's session, that today we are having a very eminent personality, Dr. Purushottam Sutar. He, by now, everyone knows through the medium of different instructions that we have been putting out different flyers, that he is the product and business development manager with Hitachi High-Tech India Pvt Limited. And he is primarily responsible for growing Hitachi's spectroscopy and liquid chromatography business in Southeast Asia. He did his PhD way back from the Institute of Chemical Technology, ICT, Mumbai, with a research topic that was based on liquid chromatographic technique. In his previous tenures, he has handled the role of a domain leader for pharma CMC market, responsible for promoting your novel LC, LCMS, MS solutions with respect to improving the user or enhancing user experience. So he has also worked with different other firms, where he has been in the role of application support, and he has excellent insight with respect to the demands and needs of the pharmaceutical industry, that I have experienced personally while I have been interacting with him when we were colleagues, and also afterwards he joined multiple firms as application scientist. I know the depth of knowledge that he has. He has especially technical expertise in liquid chromatography, like your HPLC, your IC, online HPLC dissolution interface, LCMS, MS, and also HRMS. So I guess he has substantial experience in analytical development laboratory in reputed pharmaceutical organizations, including Cipla Pharma, where we have worked together. So it's a great pleasure to have him on this session. And I had promised him that I won't take much of the time, so I took only 3 minutes in introducing him. It's all over to you, sir.
Yeah, okay. Thanks, Sujit, for this introduction, of course, and thank you for reconnecting with me after many years. Okay, it's good to meet you again. And yeah, quickly let us start with the session. Let me share my screen. Please confirm. Okay. Yeah. Hopefully my screen is visible. Visible now?
Yes, now.
Visible, right? I'll put it on the presentation mode. And yeah. Okay, so let me begin. No need to introduce again myself. I'll begin with the topic. So the topic for the day is the high performance solutions for amino acid analysis, with a little bit focus on the pharmaceutical applications. So I'll run through first the basic principles, then I'll cover up some instrumentation, obviously from Hitachi High-Tech, and then we'll see some applications as well. Okay, so again, amino acids basically, it goes to the origin of life. So those are building blocks for proteins. Protein, as we know, are essential for nutrition. They're very important for the different bodily functions, and proteins basically you break down to peptides, amino acid, and you have different studies like genomics, transcriptomics, proteomics, based on what you are studying, metabolomics as well. Then going further, these are some of the processes for which amino acids are important indirectly. I mean, they make a protein and then they help in break down food, grow and repair the body, make hormones, etc., etc. So these are some of the functions why amino acids are important. And going into the structure, basically, amino acid is a term given for a compound which has these two groups, amino group and carboxylic group. So on the left hand side, let me go to the pointer. Yeah. So on the left hand side, you see this is a typical structure of an amino acid. This is a well known thing. And you have various amino acids which differ in the R part, which is the alkyl group, or I mean, it's an organic part or a functional group. It can be a functional group as well. And you can also have some amino acid related compounds which also show the amino group and carboxylic group. Okay, so basically these two groups are there. You can classify them into amino acid related compounds as well. Now, again, there are various classes of amino acid based on the R. What R content you have. They can be aliphatic, hydroxyl, sulfur containing group, cyclic, aromatic, and there are in numbers. Okay, so basically you have the protein which comprises our body. Those are naturally occurring 20 amino acids, but amino acids area doesn't limit to those numbers. It can extend also to the non-naturally occurring or amino acid. It can go beyond 40 or even more. Okay, so let us see about in detail. And this is just a classification where from the 20 naturally occurring amino acids, there are certain which are essential. So those are absolutely required for your bodily functions, and there are non-essential amino acids. Not going further deep into this, this is an important chart because there is a classification again of amino acid based on what is the structure. So amino acids with electrical charge side chains, amino acids with polar uncharged side chains, some special cases with sulfur or different structure. And then there are amino acids with the hydrophobic side chain. So if you see broadly, this would be polar, the top one. Middle would be a little bit neutral, or the mid polar compounds, and this would be typically having a non-polar character. Okay, these are very important because this will impact on how you do the analysis of amino acids. Okay, again, as I said, there are many amino acids apart from the 20 naturally occurring, and it can, I mean, you can, I mean, there are greater than 900 reported non-proteogenic amino acids as well. So earlier one was the proteogenic, and these are the non-proteogenic amino acids. Going to the principles, it dates back to 1958, where these two scientists, Moore and Steen, they invented the ion exchange chromatography for analysis of amino acids. They basically developed a method which is a post-colon nin-hydrin derivatization method. We'll talk about this more in detail. It can be used for both qualitative and quantitative analysis. And the good part about this is, since its inception, this has become the international standard or the gold standard for amino acid analysis. Little bit going back into the basics, when I talk about chromatography, it is one of the methods for analysis which involves prior separation of the compounds of your analytes followed by the measurement. And based on the separating entity and the entity which carries the analyte over the separation medium, you can have a liquid chromatography, gas chromatography, or supercritical fluid chromatography. Okay, the technique that has been used for amino acid basically falls into the liquid chromatography. Okay, going further, Before going into the depths of it, let us understand some basic points like what is a mobile phase. So you need so for analysis on a chromatography, you need two entities. One is a mobile phase and stationary phase. Stationary phase is the one where the separation takes place, uh, and mobile phase is the entity which carries the analyte over the stationary phase, and then you will have end result is a chromatogram where you see each target peaks has a target analyte as a peak represented as a peak. So if you have one analyte, you will have one peak. If you have 10 analytes, you will have 10 peaks. So this is just a brief description of what we'll be talking about. And what you need to do is the area under the peak is used as a quantitation tool, and the retention time, the apex of the peak, is used for identification. So that's where this concept is very important. uh, how the separation happens in any chromatography: you take a mixture of analyte, you put it on the start or initial part of your separating media. In this case, I will talk about it as a column, because it's a cylindrical shape in which you fill your stationary phase. So at the inlet of the column, you put your analytes, and then your mobile phase goes, shown by the direction of the arrow, and there are separate interactions. These interactions we'll talk about more in detail. Due to differential interaction of the analytes with the stationary phase and the mobile phase, you have them separated over a span of time, which are then represented as a column, as a chromatogram. Sorry. Okay, and uh, based on the separation mode, you have different types of chromatography: normal phase, reverse phase, ion exchange, size exclusion, affinity chromatography. So we'll restrict our discussion to the ion exchange chromatography, which is being utilized for analysis of the amino acids. Okay, so ion exchange chromatography is nothing but it is based on electrostatic interaction of a charged analyte and a charged stationary phase. Okay, so here basically you will have a cation exchanger, which is shown here in this diagram. So if your cation exchanger or a stationary phase is a positive entity, then it is attracting the negative ion. Okay, opposites attract, and the remaining entities with the similar charge, they flow through the column unretained. So again, the retention of the analyte which happens depends on the charge it carries. It is a single charge, double charge, what is the atomic radius or the atomic weight of those compound, or it can be a molecular size or molecular radius. Depending on that, it will definitely be retained at different points on the column. And then what you do is, once you have retained everything, you change the effluent strength or pH so that it can then remove it from the stationary phase and give you a sequential illusion of those analyte in the form of peak. Okay, this is a very basic way of how ion exchange chromatography works. Okay, and this is exactly which is used for protein, peptides, amino acid analysis, because these are nothing but charge analyte. Okay, so going further, yeah. So the retention. In this amino acid analysis, basically the retention mechanism is ion exchange chromatography. Here you usually have a sulfonated divinyl benzene polymer as a stationary phase, which basically attracts the positive charge. So you have sulfonic acid groups which ionize, and then they have a negative charge, and then your analyte, which is amino acids, they take up a positive charge and become positively charged species, and then they start interacting, and you have the retention and elution. Okay, so uh, so basically what we do here is, when you load the amino acids onto the column, except the very acidic one, everything's are retained on the column, and the acidic ones they elute out first, followed by the neutrals and the basic ones. So this is basically how the amino acid analysis works. uh, yeah, going forward, this is exactly how it is done. In an acidic solution, your amino acid, the NH group, ammonia group, takes up the positive charge in acidic eluent. It then is retained. Once you come to a neutral solution, a neutral eluent, then your charged are neutralized. Then the retention is also governed by the other part of the structure. and eventually in the basic solution you have a amino acid which is completely in a carboxylate ion form, and it has absolutely no retention on the stationary phase. So this is how you control your uh, retention and elution of your analyte, and these are basically done by the buffers, which are basically sodium or lithium buffers, typically a citrate buffer of both the cations, and you have to control the pH and the salt concentration to eventually make this happen. So, so in here there are basically two mechanisms that's affecting the separation in the ion exchange chromatography. One is the pH. The amino acids go from positive to neutral state, and so are released from the fixed anionic site. So you start from a low pH, and eventually you go up to the basic pH. So that's how you start. So in, if you refer this in an acidic pH, you will have a lot of retention. Unless your analyte or your amino acid is very highly acidic, it will always retain. So only the acidic amino acids, they will elute out first, and then followed by neutrals and the or the amphoteric ones and the basics one as you increase the pH. Then competitive ion exchange between the cations of the eluent and your amino acids. So if you see this, ammonia is strongly retained as compared to the lithium and sodium, and hence you select lithium sodium as the eluent, so you can control. So ammonia containing species can be retained first and then eluted as per your choice by increasing the pH or concentration of the lithium or sodium buffers. Okay, and yeah, so basically you are dealing with the when you do the amino acid analysis, if you have a protein which makes up our body, you need to first hydrolyze it into the typical amino acids. Okay, and when you hydrolyze it, you have a 20 set of amino acid, which basically doesn't need a longer runtime. So in that case, your sodium buffers will work very nicely, but you are dealing with the free amino acids apart from these 20, which can go up to 40 or even more. Then you go for a lithium based buffer uh, to analyze those to separate out those species, but it can take a little bit longer time. Okay, and this is how a typical chromatogram for the uh, proteogenic or naturally occurring amino acid looks like. So in acidic eluents, you have all the acidic ones, aspartic acid and all these. Then you have the neutral amino acids, alanine, cysteine, valine, and then finally you have the basics one, including the tryptophan and arginine. So all these things here we have nicely shown that it can be captured into within a time span of 30 minutes, and that's one of the advantage of the Hitachi instrument. So yeah, going forward, when you have to analyze what methodology you should select. Okay, of course, if you see the amino acids in itself, they have a very low UV absorbance unless it is containing a benzene ring. I think apart from tryptophan, all the other amino acids uh, so in that case, the choice of a UV detector is very less. They don't even have a fluorescence. So what you need to do is, whenever you are analyzing an amino acid, you usually go for a derivatization method. Now in derivatization method, you can either go for a pre-column derivatization, that is, you derivatize before the separation, or you can go for a post-column derivatization method. In post-column, the gold standard method is the ninhydrin method, and the other one is the OPA, orthothalaldehyde method. In this, and then in pre-column, you have similarly some reagents which can be used. So it's up to you. How do you do it? Hitachi works. It is based on the ninhydrin method. Okay. So this is just a diagrammatic representation of what happens when you actually do this derivatization. In pre-column, you have a certain chemical reaction which is to be done before the separation, before you put the analyte via your auto sampler onto the column or the separation media. Then the separation happens. So the advantage is you have many types of reaction reagents. The analysis time is short, because you are practically using a reverse phase chromatography. Hindsight is the reproducibility, and there can be a lot of impurities which can interfere with your separation. So mostly we can limit this use for qualitative work. But if you need good quantitation capability, post column is one of the best technique, because here you first separate the analytes and then you sequentially derivatize via chemical reaction to enable a chromophoric entity into the amino acids, and you go for a detection. And the advantage is this can analyze many amino acids even apart from the 20 amino acids, and it can be applied for any matrix. It can be very simple matrix like a hydrolyzed protein, or it can go to wine or serum or urine. You don't need to even hydrolyze them. You can directly analyze the free amino acids from them. Okay, so that's where it is. Then some of the features here, the most important difference between an ion exchange resin and a reverse phase silica. So if you see the pre-column method, the pre-column method generally uses a C18 or a reverse phase silica based column. Okay. Now the difference between them is the chemical selectivity. So bonded reverse phase silica are manufactured in a way that they have a monotypical chromatographic behavior. That is, they will only work on one particular mechanism, partitioning or hydrophobic interaction, but it's always one is the dominant one. But if you go for an ion exchange, it has a many interaction. So separation can be based on the ion exchange, it can be partition, adsorption, size exclusion, and many more. So a mixture of these forces really can address your analytes, which I showed in few slides back, that based on structure you will have the polar amino acids, neutral amino acid, basic amino acids, and all sorts of structures are there. The charges are there. So it is really a little bit challenging to separate them on a monotypical reverse phase column. Again, on ion exchange chromatography, the retentions can be controlled by any single parameter like concentration, pH, column temperature, and you can really have a more degree of freedom on the separation. Okay, so it gives you a lot of flexibility to develop a very rugged method for the amino acid analysis. Then, although pre-column derivatization is popular, of course, because you can use the same HPLC as a for other applications as well, but it limits its use to the simple protein analysis, protein hydrolyzed analysis. When it comes to a very complex matrix, sorry, yeah, complex matrix like carbohydrates and organic acids in wine, fats, tobacco, or fats in tobacco, or macromolecules in urine, serum, or urine, then you really need a very good technique, rugged technique, which post column can provide. Other than that, no combination of pre-column chemistry and reverse phase chemistry has been able to resolve the full complement or full set of amino acids. And in reverse phase chromatography, all species in the solution, they compete simultaneously for derivatizing reagent. And this is very important because when you do it as a pre-derivatization, you have the entire bunch of your analytes. You may have 20 amino acid, 50, 60 amino acid. You just put the reagent there, and then the derivatization efficiency will be influenced by the concentration of all the solutes involved, including the matrix as well. So the efficiency of derivatization sometimes get hindered, and you may get a non-reproducible result. So, but that's not a problem with post-column derivatization, because you first separate out the analyte on the column, then there is a sequential elution, and then you let your derivatization reagent enter, and you do the reaction. Okay, so it's very straightforward. Apart from that, because you are not sending the derivatization agent onto your separating column or the stationary phase, you do not affect the chemistry of the separating phase. So your life of your column really increases when you use a post column derivatization. Okay, so yeah, that's what I said. So post column method avoids unnecessary loading of derivatization agent onto the analytical column, enhancing the life. no challenges to separate the derivitizing agent. So once you put your derivitizing agent along with your amino acids, you need to also separate it out from your analyte. Okay, that's another challenge. So chromatography and derivatization are two distinct and separate events which can be optimized independently, and you can have an optimum use of reagent because it's also costly in terms of the quantity, how much you use, and it can be also time programmed to in fact select to detect a selected amino acids. So there will be no background interference, no impurity interference here. So basically it is a more rugged and repeatable than pre-column analysis. Okay. The other thing is the sample prep is very simple. So if you see on the left hand side, it's a pre-column method where you will have your orange juice. I mean, it's selected here. You need to have multiple step of pre-treatment. But important point here is whatever you do, you need to neutralize the reagent. If you use an acid, you have to neutralize by base, because all those content you may be sending it to an LC or for that matter even a mass spec, which has its own challenges. Okay, but here the methods are very simple. The stationary phase that you use is so good that it can tolerate the acidic reagent as well, or the diluent as well. So there are really bare minimum step, and you can straight away go for the analytical work. Coming to the reagent here, because I'll restrict it to the ninhydrin one, because Hitachi Amino Acid Analyzer uses ninhydrin. So how does the ninhydrin work? Basically, it reacts with the amino acid 1:2, and then you get this kind of reagent. One is a rumen purple color, and for selected few amino acid, you get a yellow color, which they detect, which they show lambda max at two distinct wavelengths. So any amino acid analyzer works on dual wavelength. One is 570 nanometer and one is 440 nanometer for the different amino acids. So the ninhydrin reacts to the compounds and having only amino group. Then it has a high stability and highly reproducible. Okay, so this is how the reaction happens. So ninhydrin is a selective oxidizing agent which causes oxidative decarboxylation of alpha amino acids, causing to producing CO2, ammonia, and an aldehyde with one carbon atom less from the parent acid. So the next step is the reduced ninhydrin then react with the ammonia to form this compound which is called the rumen purple. Secondary amines, this was for primary amines. Secondary amines such as proline and hydroxyproline, they react to form a yellow compound which absorbed at 440 nanometer. And the reaction of an ninhydrin with the amino acid is a slow process. Hence, we need to carry this reaction at a high temperature at about 130 degrees. So that provision has to be there in the instrument. So this is how a typical chromatogram looks like. You have these bottom amino acids at 570 nanometer. On the top, you have proline and proline at around 440 nanometer. Then after separation, each amino acids will undertake a reaction at high temperature, and they need to be detected by a UVS detector. So a dedicated amino acid analyzer is able to control all the separation and reaction parameter to achieve the best reproducibility. Yeah, so this is how the Hitachi instrument looks like. Then this is a flow diagram of how this works. So here you have your pump 1 with the eluents. These eluents are nothing but buffers with different pH. And then here you have a ninhydrin reagent and a washing solution. So briefly what happens is this is the path for your eluents from the pump 1 to enter into your auto sampler column, then the reaction unit, and then detector. So here what you do is you inject say 20 microliter of your sample onto this path, and then the separation takes place here first. And once the separation happens from the pump 2, then you can send your ninhydrin reagent, and the reaction happens here as around 135 degree. The system is good enough because ninhydrin has to be kept in a very stable state. So the system here is kept in nitrogen environment, so the degradation of the reagent doesn't happen, and you can use the reagent for a longer period of time. No need to uninstall it from the system and keep it separately. It can preserve itself on the system. Okay, that's where the reaction happens, and then it is detected. Okay, to summarize this part, so basically in basics, I have covered the separation, which is the cation exchange chromatography, the reaction, which is the post column, and then the detection at 2 wavelengths. Coming to the instrumentation, so this is the history of what Hitachi has done. It was developed in 1962. It was a very big instrument with a capacity of 1 sample per day, and then over the years it has Hitachi has worked and developed this instrument eventually to a desktop model. So the first instrument was almost a room size, and then now we have a tabletop instrument called LA8080 Amino Saya, which is again based on ninhydrin method, and it can do the analysis within 30 minutes. Okay, so these are some of its features. It has a very low detection limit, 2.5 picomole, uh, with the high sample capacity of 100 samples. You can load it in the instrument, and the software is fully compliant with 21 CFR Part 11. So the data can be submitted for regulatory submissions. Okay, and yeah, it can analyze more than 40 amino acids as well. This is how the actual instrument looks like. So as with the other chromatography instrument, it will have uh, reservoir, which is again here called as a reagent box. This houses the eluents of various pH. This reagent box houses the ninhydrin reagent and a stabilizing buffer. Then you have the pumping system, pump 1 and 2, respectively, for the 2 elements, your auto sampler. Then in this part, you will have your column. After column, it goes to the reactor. This is the gas panel for nitrogen, and then you have the detector. So this is how the inside of the instrument looks like. Okay. This is the pump, auto sampler, reagent box, ninhydrin, and its buffer. This is where the separation happens. This is the reaction unit. Okay. Going further, these are some of the features here. So some of the advantageous features of Hitachi. All the reagents are commercially available, so you don't need to prepare anything. Everything is readily available. Direct plug and play. It has an automatic warm-up system, so a system can also prepare itself for user to come and then directly start the analysis. Once the analysis is complete, system can do automatic cleaning, because if you have to ensure that after your analysis, none of the ninhydrin reagent, is sticking into the lines. Otherwise, it will lead to blockages and clogging of the system. So it can smartly do the auto cleaning in an event of completion of sample as well, and if there is an error. And the entire system is maintained under the pressure of nitrogen to avoid any deterioration of the buffers and the reagents. So you don't need to separately store them. Okay, so and then Hitachi is the first manufacturer to introduce and use 3 micron column. Yeah, sorry. Yeah, it is the first one to use a 3 micron particle size column, which has enabled a runtime as short as 30 minutes and maintaining the resolution of greater than 1.2. This is the advantage of using the Hitachi instrument. And it has the concept is basically to have a comfortable operability, compliance, compact design, and high reliability. So yeah, I'll not spend much time on that. So yeah, comfortable is you have a screen where on the same screen itself you have all the options available. So you don't need to toggle between multiple screen. Everything right from operation to creation method everything is available here. Yeah, this is an important function. So there is a function for automatic reagent amount calculation. So the runtimes are long. Sometimes you have many samples, so you don't want any reagent to be get wasted or elements to be get wasted. So you can basically calculate how much is required and according to place it on the instrument. Other than that, you can also have the maintenance information just to judge the life of the instrument and periodically do the change of the consumables as well. From the legacy instrument, we have come down so that you can save a 30% of the space in your, it sits on a table very comfortably like a regular HPLC. Over the years, we have developed a lot of methods which have been proven and tested over the time, and those are, so you pick up any method, it is applicable, and easily you can transfer it to the latest model without any difficulty. Yeah, and the reaction unit which we use is called a TDE3, thermodynamic efficiency, the third. So this is basically a patented reaction column. So that this is where the reaction happens, which enables high sensitivity. Previous one was 3 picomole. You can go 0.5 picomole down. Then it can also address large molecules such as di and tripeptides also, not only the simple peptides and amino acids. Then it has an average life of 5 years. Previous generation had 2 years, but again for 5 years, if it is used properly, then you don't need to change it for 5 years, provided there is no blockages. Okay, if you maintain the system, the life is really good. And this is the heart of the analyzer, because if it doesn't work, then you do not get your good reproducibility or sensitivity. On the compliance part, you have the open lab software which is compatible here. So we ensure the complete compliance. And then we also offer ready to use method. So for a simple protein analysis, you have a protein hydrolyzation method for 20 amino acids. The entire package is there. You can straight away use it. For free amino acid, you have the physiological fluid methods. So here, again, the sample prep and everything is provided. You can use it directly. You can do not only pharma, but the nutrients, fertilizer, feed, food, biological sample, everything. So provide the support. We do have multiple columns also available for both methods. So if you are looking for a shorter runtime, you can have a standard analysis column, or 80 minutes runtime, you can have that. Even for the PF method, you can select various columns as well. So yeah, so these are the advantages which I could highlight. Has automated operations, highly stable derivatize analyte for detection, the nin-hydrogen derivatize product, long column life, highly efficient reactor for high sensitivity, very simple preparation, sample prep. You can go up to 50, analyze up to 50 amino acids, so your cost actually reduces. High stability, high reproducibility, and separation efficiency and compliance. So now that I've covered the basics and instrument as well, let me go on to quickly to the applications. I'll focus on the pharmaceutical application. However, this instrument is not limited to that. You can do food analysis or any matrix which has amino acids, or the amino acids can be generated by hydrolysis or any step or any sample prep. You can analyze here. So coming here to the pharma application, basically this is I've briefly shown a drug life cycle which involves discovery, preclinical, process development, analytical method development and verification, and stability studies, then transfer to the QC, and then post-market support. Everywhere amino acid analyzer can be used to address each stage. Okay. Then once you talk about all these stages, every stage has to adhere to the global regulatory requirements like the USP, EDQM, ICH. And if you go through all this method, all of these three regulatory bodies, they mandate the use of a dedicated amino acid analyzer. And of course, they do mention this kind of derivation technique. So use of a dedicated amino acid analyzer is essential when you are submitting your data for regulatory compliance. So some example, this is the first one, calcitonin, which we have done with as per the USP monograph. So we have broken down, hydrolyze it using acid hydrolysis and performic acid hydrolysis, and we have got the separation within 30 minutes, and the recovery of the amino acids is within the criteria of 90 to 105%. So this is a very simple peptide with disulfide linkages. Coming on to the engineered peptides. So nowadays you hear this lot of peptides being used for the obesity, diabetics. So now that area has become really hot. So what happens is these are not the in addition to the amino acids, they do have a lot of linkers or side chain. Now once you have those linkers and side chains, um, uh, you need to be ensure how accurately you analyze your sample, because these linkers and side chains are added to maybe enhance the life of the peptide, or maybe therapeutic efficacy of the peptide. So that's why they are incorporate here, to have a formulation which can serve its purpose. And then, of course, when you add all these linkers and peptides, you need to also justify them through analysis to the regulatory bodies. So we have taken up one of the well-known peptide, which is the GLP analog, anti-diabetic, the liraglutide, which is the first generation one. So once you have, so it has a linker here at C16. It's a 31 amino acid peptide. So once you do this, there are a lot of questions like, does it cause matrix interference or analysis? So this is gamma glutamine linker, which is added here. Does this affect your sample prep technique, or what would be recovery? So we did the analysis with our standard process, hydrolysis, which we have, and then we found the recovery was very good. It was about 98%. So the as such methods can be used here. Of course, we need to work on the separation for additional amino acids, and this is the separation we have achieved on within 32 minutes. Then the second generation one is the semaglutide. Again, the similar questions here, because apart from this, you have the additional groups which are embedded in the peptide chains, like the AIB and AE, AEE. So these are groups again which are embedded in the chain, and once these groups are present here, how does it affect the recovery or derivatization? All those questions are there. So this one even we analyze, and we got a nice recovery of more than 88% in for this. And importantly, apart from the regular amino acids, we were also able to separate all these non-proteogenic entities, the AIB, AEEA compounds, on the given method. Of course, then we had to increase the runtime. Yeah. Okay, then this was an experiment where all the non-protogenic entities which are used in the regular peptides like sema and liraglutide, can we have a single common method to address maximum peptide drugs? Okay, so in an attempt, we were able to incorporate these amino acids related compound in this chromatogram and get the separation. The important point here is all the analogs of leucine, alloleucine, isoleucine, leucine, norleucine, are very well separated on this chromatography using a UV detector. Now, these are also tough to first separate even on pre-column, and then have them analyzed by maybe say MS detector or fluorescence detector. So this is one of the achievement that we have, that you can actually separate all these four analogs in the single run itself. Yeah. Moving forward from the peptides, the regulators are also focusing on the use of the amino acids. So impurity is in amino acids. So once you have a purified amino acids, what are the impurities present? So again, these are adherent to the regular methods, regulatory methods. If this is done by EP guidelines. The batch A is virtually free of impurities, and the batch B shows some amount of impurities, ornithine and lysine, which can be calculated. Then there is for biopharmaceutical drug development, there is what we call as cell culture media. So basically, in cell culture media, you have an organism like a CHO cell line, which can generate antibodies or therapeutically useful moieties. So in that cell media, you need to feed certain nutrients. So amino acids are one of the nutrients. So it's very important to know the consumption of those amino acids by the organism. So here, amino acid analyzer can actually help you do a time study from 0 to 48 hours, for example, given here. you can actually see how much of what amino acids and are consumed in how much time. Accordingly, you can do the replenishment. So what happens is, if you don't monitor this, if the nutrition is less to the organism can die, and your study goes waste, or maybe you won't get the desired therapeutic product. Okay, so this is a very important study which amino acid analyzer can help you with. It can also do the analysis of immunoglobulin G protein. Again, this is shown here using the acid hydrolysis, performic acid hydrolysis, mercaptoethane sulfonic acid hydrolysis. All the hydrolysate you can analyze on the amino acid analyzer. So here in this case, performing acid analysis is actually converts cysteine and methionine to the acidic forms, which are also separated on this chromatography. So practically we cover a full set of amino acids here. Okay, so we started the peptide, then the amino acid, then we moved on to cell culture, and even large molecules like immunoglobulin protein can be done on amino acid analyzer. So to conclude, so basically you can start from discovery phase and do all the analysis, composition analysis, peptide identification, impurity, amino acid analysis. Then you have the QC, batch to batch check in cell culture media. Right from discovery to QC, you can do everything on the Hitachi LA80 amino acid analyzer. And if you want to refer to some of this information, this is a site from Hitachi called SI Navi. You are welcome to register on this site, and you can refer to the application data with respect to product, or for that matter, any other product. Okay. So with that, I conclude my presentation, and yeah, I think we are open to questions. Any. Yeah. Suji, yeah, you can take over.
Thank you, Dr. Purushottam. That was really wonderful session. Any questions with respect to what we have discussed so far?
In the chat.
Any questions once again? I guess this is the most toughest part rather than the presentation.
Yeah. There are certain questions which are not right now here, but we can certainly put in the chat, or we can address it later also, no problem.
Yes, definitely. I guess if there are no questions, we will conclude this session. I personally thank Dr. Purushottam for accepting our invitation and presenting such a wonderful session on amino acid analysis. As you rightly said, semaglutide and the category of the drugs that we are seeing, especially right up to 2030 with the patent expiry and all coming up. So this obesity drugs, and this is going to be the means it is going to be the future of pharma industry. So I guess we have one question once again before I conclude. Purushottam, can you see that question?
Yeah, I saw that. It's preparative level available. Yes, currently not with Hitachi, but there are instruments which can do prep analysis for recovery of the amino acids as well. So currently, it is not available with Hitachi, but you can certainly do that. Thank you. Not with this instrument. So this is only for profiling of the amino acid analysis. Peptide amino acid sequencer is a different instrument. So this is merely this instrument basically captures the test that is mandated by the USP and EP, that you need to profile and quantify the amino acids. So this is only limited to that. Sequencing, you need to have a protein sequencer or a peptide sequencer. Separately.
before, after you have that sequence in place, then you go for this instrument. Correct.
Thank
Isn't
you.
it?
Yeah, correct.
Yes.
So once you confirm the sequence, and then you need to break it down, and then you need to identify what has gone into the sequence. So this is added an additional to the sequence.
Any more questions?
We have 10 more minutes.
So, okay. So,
Yes, good. Thank you
since
so much.
yes, it was in fact a very good session, because the main thing about this was that we didn't you started with the basics. So who were not aware of what an amino acid is, of course, those who have registered would be aware of these things, because they understand the importance of an amino acid analyzer, and or the experimental part of amino acid analysis. That's why they have registered for this particular session. So you started with the basic concepts. You went on to explain the difference when it comes to the instrumentation part, and also the application part. So I guess fair bit of everything that needs to be covered with respect to our understanding amino acid analysis, you have covered it. So as Dr. Purushottam also mentioned, if you have any questions, you can direct it to us, or you can directly contact Dr. Purushottam also. Okay. Okay. So they won't allow me to conclude.
Yeah, how to test? I have one question. I am producing bioenzyme. How to test using HPLC? So maybe you can just enlighten us that in your final product, do you want to analyze amino acid or the entire molecule itself? How what is your final aim? Because HPLC is a very broad technique. Depending on the detectors, you can do many things. So if you can just elaborate on the question, then I'll be able to help you. But if you are asking if you can use, I mean, you can do the amino acid analysis from your bioenzyme, definitely possible. Definitely, yeah.
Mr. Ajay, are you asking with specifically with respect to what all tests needs to be done with respect to bioenzymes, or any specific test you are asking? You can also speak
Yeah.
here,
You
because there.
unmute and speak. Maybe no problem.
Mr. Ajay, are you there? Can get your question clarified with respect to your production of bioenzymes.
Protease, yeah, definitely you can do this. If your aim is amino acid analysis from this enzyme, you can definitely do this. There's no limitations here. But anyways, I think Sujit,
Okay.
the message that I wanted to basically give from this particular, yeah, sure. I will share. Maybe you can share certain information like a mail ID or something. Then I can either directly communicate to you on the entire procedure where you can analyze this particular enzyme. Please do that. I mean, I can take it from Sujit as well, and I can communicate with you. No problem. We can have detailed discussion. Yeah, coming back to my point is the whole idea of this content which I have prepared is there is always something dilemma whether to use a pre-column method or a post-column method. Okay, so the intentions was to basically if you need more accuracy and reproducibility, post-column method is a really good method due to various factors that I explained. Okay, that's something which I wanted to put across the forum, which is always a question that even we get. Why not pre-column? Why post-column? So that is something which I wanted to put across. Yeah, that's the
Okay,
thing.
done. I guess if there are any questions, we will address them directly. Okay, Mr. Ajay, with respect to the analytical or these activities, you can directly mail either Dr. Purushottam, or you can contact any of us. We will help. I guess Purushottam will be sharing his mail ID also here. So if you have any specific requirements with respect to the instrument also, he can help you with that.
So I have shared my mail ID.
Yes,
But even you can share it to the participant.
Yes, perfect. Means with respect to the instrument also, because we personally know the instrument, or the accuracy of this instrument. When we have seen the results also in previous data also. So I guess Mr. Ajay can contact you directly for that.
Yeah.
Okay.
Sure. Great.
Okay. So thank you each one of you, especially taking your time on a weekend. Special thanks to Dr. Purushottam. We will see you in the next session with yet another wonderful topic. And as Purushottam also already mentioned, if you have any specific queries, don't hesitate to ask, whether it is specifically towards Hitachi Instruments or in general. I know he is a very he won't hesitate to respond to you. Okay. Thank you so much. I will be sharing this recording on YouTube channel also for ready references. So once again, a happy New Year, everyone, and happy weekend. We'll connect during the next session also. Okay. Thank you so much.
Thank you, Sujit, for inviting me again. Thank you.